This is the short video of the mitochondrial replication DNA damage and repair. So in that section, I'll just in brief what about the mitochondrial genome and all those things. So you all know mitochondria are present in the eukaryotic cell. So it's kind of semi autonomous organism, and along with the double stranded DNA is present in the mitochondria. It's a genetic material of that double stranded DNA. Along what we have seen, it's a powerhouse of the cell because the ATP is produced on the mitochondria. So basically mitochondria that can produce ATP. So what are the mitochondrial genome? In brief, it's a double stranded DNA. In that also the G-reach strand. The G-reach strand is a heavy strand, and another strand is a C-reach strand. C-reach strand is a light strand. So if you take one circle of that, because it's a circular DNA, so you can assume that the one strand is full of the G means guanine, and another is the C-rich cytosine. So linear mitochondrial DNA can found also. It's found in the green algal genus called the Polytomela, and it's a chlorophyceae plant. So in the chlorophyta, so in that plant mitochondria is linear DNA is there. So you can assume that it's a human. If you can assume one mitochondrial genome, it will be like the 16,569 base pair of the human mitochondrial genome. So if you isolate the mitochondria and then isolate the DNA of that mitochondria, then you can if you do some sequencing, that can give you the 16,569 of the base pair. So what are the key factor? You have to remind on that. Some D loop is there. It's a non-coding region of that, and it's if somewhere it can be a triple strand. So you can say about the D loop. Along with that, you can also important things like heavy strand and light strand. That's fine. The light strand replication origin outside the non-coding region. So in brief descriptions of the mitochondrial genome, it overall chapter that talk about the mitochondrial replication of the and along with the DNA damage and repair. So in next lecture I can also give some of the details about the mitochondrial D loop replication, how it is formed the D loop. So it's a non-coding region has the origin for the heavy strand, and origin for the light strand is the outside of the NCR. NCR it means the non-coding region. So origin will be the in light chain and it is in the NCR region. The triple stranded displacement D loop structure is formed by the premature termination of nascent H strand DNA. H means the heavy strand DNA synthesis as the termination associated sequence and also known as the TAS. So in complete brief, all the time I'll give the brief of that everything. So it's like you have to remind about that TAS region. TAS means the termination associated sequences. Now you learn about the NCR non-coding region, and you learn about the D loop. They are the triple stranded displacement, or that's called the D loop. And also DNA polymerase is here the gamma. It's had a 3 subunit, cannot use as a double stranded DNA as a template. So you have to remind the single point also. And helicase is there, that is twinkle is there, twinkle, that's the also name of the helicase. Also they are continuous synthesis for the both stranded, and it will be like the unidirection from each origin, like the other bacteria that whatever their unidirection on that origin, and the light strand promoter also there in that region. So we learn about the TAS region, termination associated sequence region. We learn about the NCA region, and we learn about the LPS, the light strand promoter, and these are the all key factor found in the D loop section. And replication always initiated in the OH to produce the new heavy strand. Initiation of the replication occurs through the synthesis and RNA primer from the LSP. LSP means uh, light chain promoter region and forms the G quadruplex with non-template DNA and terminates prematurely. So we have to remind all those easily. So DNA primer is not used. The RNA primer is used for the mitochondrial DNA replication. Hope that this will be long thing. We have to remind 2-3 molecules like MTSSB, MTSSBP1, and along with some names like you have to remind about the polymerase gamma. I'll discuss with the later videos also what are the gammas and replication continues with the polymerase. And if you need to know about all those things, I can also discuss within that figure, but I think that is an also medium to talk with that, like I can talk with you about that mitochondrial DNA and all those related things, and it will be helpful to you.